Hello everyone. I'm doing this video recording for our infection PowerPoint that we didn't get through yesterday. Labor Day is kind of messing us up for sure. But anyway, it is not as long and involved as the inflammation one. So I'm going to get through this with you so that you have somewhat of an understanding a little bit more about how we look at infection in pathophysiology. And you have the very nice PowerPoint to look at. So go ahead and do that. I took the pictures out because it's much easier to save. It doesn't take up as much room, but it's the same information. So here are our objectives. And then there's just a little picture of the chain of infection. We know that there's certain things that have to happen with whatever the bug is, a parasite, bacteria, virus, a tick, whatever it is, we know that there's got to be a way in and a way out. And how do they replicate? They're all a little bit different. So let's look at some of this. This slide is just giving you a little history and some statistics. TB is the leading infectious cause of death worldwide, still killing roughly 1.6 million people in 17. In the past 200 years, it's claimed the lives of more than 1 billion people. More deaths than if you total all these together: malaria, influenza, smallpox, HIV, AIDS, cholera, and plague combined, and COVID-19, simply because it's been around longer. But COVID-19 is on its way. We know that we're living it. It's been a nightmare. Infectious disease is a significant cause of morbidity and mortality worldwide, and we are called in the nursing world to try to halt this in every way we can. Microorganisms find the human body to be hospitable. It works. We have the adequate nutrition. We have the right temperature. Remember, warm, moist, and dark. There it is. Microorganisms and human beings. Normal flora, I talked about this quite extensively yesterday, so you can just read through this. We know where it's found. It's on all the openings, basically, of the body. Skin contains 10,000 microorganisms per centimeter squared. You don't need to remember that number. It's just kind of a reminder as to really, skin is an organ, and it's an excellent organ, serving as a barrier, amongst other things, to the rest of our organs. Trillions are secreted from the GI tract daily. Intestinal flora also helps with vitamin K, synthesizing vitamin K. We give vitamin K to infants because they're born with a sterile bowel. So until the intestines have some food in it, the vitamin K is not synthesized. And a lot of nurses mix that up because a lot of clotting factors are synthesized in the liver. They say it's because their liver's immature, but it's not. It's because the GI tract is sterile. And that first black meconium stool that they get that looks like black tar actually has no smell to it at all because it's sterile. But anyway, just another important point of where microorganisms are or are not. Pathogens have been able to change to overtake barriers. We know that. Some bacteria produce thick capsules of carbohydrate or protein. And this, because they're so thick, they prevent our phagocytosis. And remember us using that word continually on our last lesson. Because we need that. We need that process of phagocytosis. Some produce toxins that kill neutrophils. Now, what do we know about neutrophils? We know that they're the first white count that shows up. Some pathogens proliferate faster than the immune response can attack or fight it off. Remember how it said something like it takes 4 to 6 hours roughly for a neutrophil to show up, and then 24 hours later, we have our mature macrophages. So during this whole time, this pathogen is wreaking havoc in our body. Viruses are sneaky devils, just like our cancer cells. They hide inside cells. They're intracellular parasites. However, when moving from cell to cell, identified antibodies can block the virus from neutralizing the antibodies. But we have to have antibodies first. That was our problem with COVID-19 last year. And this year, too, if people aren't vaccinated or they haven't been exposed, is that they don't have an antibody. We did not have antibodies because it was a new virus for human beings. and that was the problem. We couldn't block it. Some viruses change their surface antigens all the time. And the influenza or the common cold and that flu, sometimes it's not just a cold. You have aches and pains and a fever and bad cough, and it can change every year. And that's why the flu shot changes every year. That's why they say to get it every year, especially if you're in healthcare, because we don't want to make other people sick. And also, this is why flu shots sometimes don't work. They miss the mark. The public health department missed the mark on how they're producing the serum, and the viruses can work their way around it. Infection-causing microbes, viruses. We'll start with those. They enter the host cell and use their metabolic processes to proliferate and cause disease. They can alter antigenic properties of the host cell. They transform the host cell into cancerous cells and promote bacterial infection. So viruses are really horrific. Again, I'm going to say that again. They enter the host cell and use their metabolic processes to proliferate or grow, replicate, and cause disease. They alter antigenic properties, so that has to do with the antigens of the host cell. They transform host cells into cancerous cells sometimes and promote bacterial infection. They elude the immune system by making small changes to the genes that produce viral surface antigens, a process known as antigenic variation. AIDS, acquired dysfunction of the immune system and caused by a retrovirus, HIV, that infects and destroys CD4 and lymphocytes, or our T helper cells. We know that AIDS does that. Viruses must reach the cell and attach to it before it can go inside and reproduce. So now that we know that, we may be able to go get it before it gets in there. So here, let's see, what do I have here? Microscopic genetic parasites that contain DNA and RNA. They're much smarter than bacteria. Bacteria do not have DNA and RNA. They have no metabolic capability. They need our host cell to replicate. They occur when normal inflammatory and immune responses fail. The virus develops in this cell and hides there. After introduction into that host cell, the inner capsule releases genetic material causing the infection. Some surround the host cell and preserve it. Others kill the host cell on contact. So if you have antibodies against a flu virus, the antibodies can bind to the virus outside the cells and keep it from attaching and entering the cells. That's the whole idea behind that vaccine. That way the virus will not be able to reproduce, because remember, it's got to get inside the cell to reproduce, and it will not make you sick. So this, let's see if it'll work for me here.
So let's say that this guy has the flu, could be any flu, and here's a droplet from his sneeze containing, if we move in and take a really close look, you see each one of those little purple things is a virus. And there are a lot of viruses floating through the air, some of which go inevitably up this unfortunate man's nose.
How did that guy feel when you ripped off half of his face?
It was interesting because we did it while he was sleeping.
Okay, I am talking with medical illustrator David Belinsky, who designed this video for Xyrus, a research company. So here comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things. You call these keys, right?
Those are the keys.
Yeah, this is a key, this is a key, this is a key.
Okay, if the keys on this virus happen to fit the locks, which are those little purpley stick-uppy things on the surface of the cell, if there is a match, the cell, watch this, welcomes the virus in. And what's this?
This is the welcoming committee. They all interlock with each other, and they pull this membrane down into the cell.
And down it goes deeper and deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses in those little noodly things. So this unsuspecting cell has been tricked into guiding these virus recipes right into its own command center, the nucleus. So in they go.
And they are immediately recognized by this big pink molecule, which is a mini factory.
Yeah, what is it doing?
It threads the nuclear material, the instruction code of the virus, through one hole, and out another hole comes a brand new instruction set.
So it's a copying machine making copy after copy after copy of virus recipes, which then go out of the nucleus to little chefs, those blue peanutty things. They cook up proteins that go back into the nucleus where they are reassembled into baby viruses, and then out they go. They get covered up and head to the surface where they get new keys, and then boom! Here they come. This is an eruption of virus after virus after virus after only one virus entered the cell. But how many came out? Well, millions. Millions. So if one virus can produce 1,000,000 babies and do it again and again and again, how come this guy doesn't just drop dead, I don't know, in like 10 minutes?
Well, because you have about 100 trillion cells.
I see. So 1,000,000 viruses is just a drop in the bucket when you have 100 trillion cells. And anyway, remember, you do have your own immune system, which when it sees a virus, usually kills it. So while the virus does multiply fast, with any luck, your immune system will work just a little faster. So, yes, all viruses want to spread, that's what they do, but most of the time we do keep them in check. Most of the time.
Okay. There we go. Oh, God. Well, I think it ended. Sorry. Let me see. Yeah. Okay, I guess it is still recording. I thought it had ended. So anyway, interesting, huh? I love many things about that, is that we do have an immune system that can stop it. And you can kind of see they reiterated that it has to get into the cell, but if we have an antibody there, it will not let it in. So anyway, I'm going to stop here, and then I will start with another one. Hopefully finish this one. We may have to have two more. So I'll be right back.